Now we introduce that recombinant DNA into the host cell. What happens inside the host cell? Multiplication occurs. Multiplication and selection of clones occurs.
Yes, ma'am.
Okay. Then protein encoded by clone gene. This is the basic principle of recombinant DNA technology. We discussed with a diagram. Last
Yes, ma'am.
class we have a diagram of this donor DNA, plasmid DNA, and what happens all. Okay.
Yes, ma'am.
Then now we enter into, we discussed only the first part, the enzymes involved in recombinant DNA technology, mainly two enzymes, that is restriction endonucleases and DNA ligase. Okay. Then next we are entering to vectors.
Yes, ma'am.
Okay. So vector, in order to introduce the human gene into bacteria, okay, we are medical students, so we are very interested in how we can introduce human gene into bacteria for the production of vaccines and all. Okay.
Yes, ma'am.
So in order to introduce human gene into bacteria, at first the gene is transferred to a carrier. Okay.
Yes, ma'am.
That particular carrier or cloning vehicle is known as a vector. Okay.
Yes, ma'am.
So plasmids are vector. Plasmids
Yes,
are
ma'am.
vector. Most commonly used vectors are plasmids. Okay. Then what are plasmids? Very familiar with that particular word, but we don't know what is it. What is plasmid? Huh?
It they act like vectors. They are circular DNA.
Yes, circular DNA. Actually, they are extra chromosomal, double-stranded and self-replicating DNA molecules seen inside the bacteria.
Yes, ma'am.
Okay. So actually they are present inside the bacteria, but they are extra chromosomal.
Yes, ma'am.
Okay. They are circular, double-stranded, and that is the importance of plasmid. They are circular, extra chromosomal, double-stranded, and self-replicating DNA molecule, which is seen inside the bacteria.
Yes, ma'am.
Okay. Then next point is very important. In nature, plasmids confer antibiotic resistance to host bacteria. Okay.
Yes, ma'am.
Got the point? So we discuss all things. We have a donor DNA and we'll select a piece of uh, desired DNA piece from that donor DNA with the help of restriction endonuclease. Okay. And then we place that desired DNA piece one side. Then next we need a vector. What is meant by vector?
Uh,
Sandhya?
yeah, vector means
Hmm.
they introduce the human gene into bacteria. Uh,
Okay.
and they are commonly used as plasmids.
What are plasmids?
They are extra chromosomal double-stranded self-replicating DNA.
Okay, circular DNA. Okay.
Yes, ma'am.
Then, what is the importance of this plasmid? They have a special capacity.
Yes, ma'am.
What is it?
They can act like antibiotic resistance.
They can confer antibiotic resistance. That's an important point. We will enter into that particular point. Okay.
Yes,
So,
ma'am.
this feature has profound significance in clinical practice because antibiotic resistance property is exchanged between bacteria.
Yes, ma'am.
Okay. Then one of the most important thing about plasmids are present extra chromosomal. Okay.
Yes,
It's
ma'am.
a circular double-stranded self-replicating DNA. It is present as extra chromosomal DNA. So
Yes.
plasmids replicate independent of bacterial DNA.
Hmm.
Hmm? Clear?
Yes,
Uh,
ma'am.
there is no role between bacterial DNA and plasmids.
Yes,
Okay.
ma'am.
Foreign DNA could be incorporated into them by using specific RA. It is clear?
Foreign DNA could be incorporated. Yes, ma'am.
Foreign DNA or donor DNA,
Mm.
okay, desired piece of DNA that could be incorporated into them by using specific RA.
Yes, ma'am.
Okay, we know that. How can we introduce? Actually, plasmid DNA cut by restriction endonuclease, that is the term. Hmm?
Yes,
We
ma'am.
know the joining is done by which enzyme? DNA
DNA ligase.
ligase.
Yes,
Okay.
ma'am.
Then, which is the most commonly used plasmid? Most commonly used restriction endonuclease is EcoR1.
Yes,
Okay,
ma'am.
like that, one plasmid vector is there. Most commonly used plasmid vector is there. It is known as PBR322.
Yes, ma'am.
Okay, you just by heart that name. Okay, and forget about all other vectors. Okay, PBR322 of E. coli. We are
Yes,
dealing with EcoR1. So, what is the which is the plasmid vector used in E. coli? That is the most important thing. So, PBR322, right?
yes, ma'am.
Okay. Then next is one more particular thing that is used. There are some other vectors also present, which are they bacteriophages and cosmids.
Yes, ma'am.
Okay. Now we are dealing with vectors. There are three categories. First category we finished, that is plasmids. The next one is bacteriophages
Bacteriophages,
and cosmids.
cosmids.
Okay.
Yes, ma'am.
What is the difference and at which time we need this bacteriophages and cosmids? Okay, plasmids can accept only about 6 to 10 kilobase pairs long foreign DNA.
Yes,
Okay,
ma'am.
Hmm? Like our memory card. Okay,
Mm.
the capacity of memory card is different. Yes,
Yes, ma'am. Yes,
like
ma'am.
that. Plasmids have can accept only about 6 to 10 kilobase pairs of foreign DNA.
Yes,
Okay.
ma'am.
If a DNA segment of 10 to 20 kilobase pairs is to be introduced, what happens? Plasmids cannot carry.
Yes,
Okay.
ma'am.
10 to 20 kilobase pairs cannot be carried by this plasmids. So we need another carrier.
Yes, ma'am.
Okay. So we use bacteriophages. Hmm?
Yes, ma'am.
Bacteriophages may be the vectors of choice that working with genomes of.
Yes, ma'am.
Then what is this bacteriophage or define bacteriophage? Bacteriophages are viruses that replicate within the bacteria.
Yes,
Okay,
ma'am.
got it? What are
Yes.
plasmids? What are plasmids?
Uh, Plasmids, they act as vectors.
Mm.
They are used as uh, they have the
Hmm.
capacity of to accept 6 to 10 kilobytes of foreign DNA. They
Hmm.
uh, they they can
Not
be
by, it is DNA. Okay, kilo base pairs. Okay.
sorry, yeah,
Huh?
kilobase pairs. Yeah, sorry.
Okay.
They're in order to introduce they can be used as antibiotic resistance.
Okay. Okay. Then what is the importance of bacteriophage when it is come into action?
When it is more than 6 to 10 kilobase pairs, the plasmids cannot be used. That time bacteriophages are used.
Up to 10 to 20 kilobase pairs. It's also very
Ah,
important. Okay, up to
yes,
20 kilobase pairs, we can use this bacteriophages. Okay, what is the
yes.
definition of bacteriophage?
Uh, they are viruses that infect bacteria.
That can replicate within the bacteria. Okay,
Ah,
for example, lambda phage.
yes, ma'am.
Okay, lambda phage DNA is linear, 40% of genome is non-essential. There is therefore in this region we can introduce a large fragment of foreign DNA. Okay,
Yes, ma'am.
the very common in case of plasmid, we told it is PPR 322, like that. Bacteriophage, the most commonly used bacteriophage is lambda phage. Okay,
Yes, ma'am.
why we use this lambda phage is that 40% of genome is non-essential in that particular lambda phage. So we can easily introduce our donor DNA or foreign DNA into that space.
Okay.
Clear?
Yes, ma'am.
Then next is about cosmid vector.
Mm.
Okay. What is the difference between plasmid, bacteriophage and cosmid? That is the step. Okay.
Yes,
Cosmid vector that can take up still bigger fragments.
yes, ma'am.
Okay, we can use up to 20 kilo base pairs. We can use bacteriophages. Then more than 20 kilo base pairs, we can use this cosmid vectors.
Cosmid. Uh,
Okay,
yes,
20 to 50 kilo base pairs length.
yes, ma'am.
Okay, cosmids are plasmids that contain DNA sequences required for packaging of lambda DNA into phage. Okay,
Yes, ma'am.
uh, complicated sentences we can avoid. Okay, don't worry. Cosmid vectors can take up still bigger fragments of 20 to 50 kilo base pairs length. Actually, cosmids are plasmids, okay, that contain DNA sequences required for packaging of lambda DNA into phage.
Yes, ma'am.
Okay. Okay, that's about our bacteriophages and cosmids. Now we finish one topic. That topic is vector.
Mm.
Okay. Uh, Clear with vectors?
Yes, ma'am.
Okay, no doubt.
No.
Hmm? Okay. What are cosmids?
Cosmids, they are type of vector
Hmm.
which can take bigger fragments of DNA,
Hmm.
that is 20 to 50 kilo base pairs in length.
Hmm.
And that they contain DNA sequences required for packaging of lambda into the phage.
Okay.
Lambda
Then,
DNA.
okay, then we are entering into, we just say that there is a restriction endonuclease and we join with ligase and all, then we produce one recombinant DNA. Okay, that's all. But actually, how can we prepare? What is the procedure of DNA recombination that we are going to discuss? Okay. Hmm?
Yes, ma'am.
So, it's a little bit difficulties, okay? It's complicated one. Okay, don't worry, we can cut into pieces, then we can discuss, okay? So,
Yes.
first one, preparation of specific human gene, okay? We
Mm.
introduce a human gene fragment into bacterial DNA. That is our purpose.
Mm. Yes, ma'am.
Yes. So, isolation of a specific gene from the human DNA is a laborious process. Beta globin gene is about 1.6 kilo base pairs long, which is represent only 0.00005% of total human genes. This is like searching for a needle in a haystack.
Mm.
Okay,
Yes, ma'am.
it's a very difficult process.
Mm.
Okay, so we have to find out that very small part from a huge part.
Mm.
Yes.
Yes, ma'am.
So how can we find out this? This problem is generally solved by the preparation of complementary copy of that particular DNA. Okay.
Okay.
Uh,
Yes,
this
ma'am.
uh, this branch is known as molecular biology because they are dealing with fine, small particles.
Yes, ma'am.
Okay, and they are very much interested in that fine particles. Okay, that's why it is most developed area. Okay,
Yes, ma'am.
molecular biology. So many researches are conducted and PhDs and all things are in molecular biology. Okay, so
Yes, ma'am.
it's a and also the PhD scholars working in this small areas and preparation of cDNA and all. So how can we prepare a specific human gene? Isolation of specific human gene is very difficult process. Okay, this problem is generally solved by the preparation of cDNA, complementary copy of DNA. That is known as
Okay.
cDNA. Then I
Yes, ma'am.
will always tell you next time onwards cDNA. What is cDNA?
Complementary copy of DNA.
How can we produce? How can we synthesize? What is the reason for the production of this complementary DNA?
Because preparation of specific human gene is very tough. It cannot be. It's very laborious process.
Okay.
Uh,
So that is the first step, preparation of a specific human gene by this cDNA preparation. Okay. Hmm?
yes, ma'am.
You can see a number of steps in the case of the preparation of cDNA. Okay.
Yes, ma'am.
We will come into it. Okay, don't worry. It is easy to start the work with mRNA, not
Okay.
with DNA. We start our work with mRNA. In
Yes, ma'am.
replication stage, in replication, transcription, translation stage, we start our work with mRNA.
Okay.
Okay, why we work with mRNA? Because DNA that we will discuss, it is easy to start the work with mRNA. There is a selective expression of a particular gene in certain tissue. For example, insulin mRNA will be abundant in the beta cells of pancreas.
Yes, ma'am.
Okay. Another advantages of using mRNA is that RNA will have only coding sequence. We are very clear with that point. Yes or no, Sandhya?
Yes, ma'am.
Why?
Yes, ma'am.
Why? Hmm?
Because during splicing, all the
Hmm.
introns are removed and exons are there, only there.
Okay, exons are joined together. Splicing mechanism is there. Yes.
Yes,
Then,
ma'am.
in case of replication, we know DNA repair mechanism is there.
Yes,
Okay.
ma'am.
After replication, DNA repairing mechanism occurs. Then transcription is done. Then formation of mRNA occurs. Then mRNA undergo post-transcriptional process or
Yes, ma'am.
processing of mRNA. So after processing of mRNA, we get a packed mRNA with full of coding sequences, right?
Yeah, yeah,
Right.
yes.
Exons are only exons. Coding sequences are there. Non-coding sequences are cut out. Okay.
Removed. Yes, ma'am.
Okay. So from the specific mRNA, the cDNA is produced. So,
Yes,
which
ma'am.
is the mechanism which that is why we produce DNA from RNA?
Uh, uh, Reverse transcriptase.
Reverse transcription. Okay, the
Yes,
enzyme used is reverse transcriptase. We already discussed that topic. From
yes.
from
It's.
RNA, we
Uh,
synthesize DNA.
mm.
Yes. Okay. Now you got the idea.
Yes.
Okay,
Yeah, yeah.
we
Yes,
have
ma'am.
in diabetic patients, they have insulin injections. Okay,
Mm.
how this
Mm.
insulin is produced? How this insulin is produced in a large amount? We collect all insulin from human cadavers and all? No.
No.
How is
Yes.
produced? Insulin is produced through this
Beta.
recombinant DNA technology. Okay,
Okay.
that
Okay.
particular insulin gene of human being is inserted into bacteria. Hmm?
Okay.
Okay,
Yes, ma'am.
sorry. So, for example, insulin. For the synthesis of insulin, we use that particular protein. When insulin is a protein, okay.
Mm.
So we use that insulin mRNA. Hmm?
Okay. Yes, ma'am.
mRNA for this recombinant DNA technology. Okay.
Yes, ma'am.
So it's a very laborious process. So we convert that mRNA into cDNA. What is meant by cDNA?
Complementary
Complementary
copy of
DNA.
cop. Yes, ma'am.
Okay. Then we can easily complementary by the preparation of complementary DNA, we can easily clear hmm, that
Yes, ma'am.
problem. Okay. Human gene, introduction of human gene. Okay, Sandhya?
Yes. Yes, ma'am.
Okay. Okay. So I think you got the point.
Uh,
Yes or no?
yeah, yeah. Yes,
Yes.
ma'am.
Okay. So it is easy to start the work with mRNA. There is a selective expression of a particular gene in certain tissues. We know certain tissue, there is an expression of a particular gene. For example, insulin. Insulin is present in which area?
Beta cells of pancreas.
Alpha, beta, gamma, delta is there. So beta cells of pancreas that contain insulin, we take that mRNA of insulin.
Yes,
Okay.
ma'am.
Uh, for the protein synthesis pathway, we take only that mRNA. Hmm?
Mm.
Insulin synthesis, you just imagine there is requirement of insulin synthesis. So DNA undergo replication, then transcription, then translation. That particular insulin synthesis occurs in our body,
Ah,
right?
yes,
So
ma'am.
in that pathway, we take only that mRNA.
Okay.
Okay. Another advantage of using the mRNA is that RNA will have only coding sequence since introns are already removed by splicing process. From the specific mRNA, the cDNA is produced by using, which is that enzyme?
Reverse transcriptase.
Okay, reverse transcriptase. Okay. So before then we can enter into this huge pathway. Okay. Total RNAs are isolated. We can't find out the mRNA first. So for that purpose, we collect all types of RNA. Which are the different types of RNA?
mRNA, tRNA, rRNA.
Okay, so tRNA that is involved in our which purpose? Translation.
Translation.
Okay, we know that clover leaf structure and
Ah,
amino acid is carried along with this tRNA and all. Okay,
yes, ma'am.
so what happens? Here we have mRNA and rRNA.
Yes, ma'am.
Okay, tRNA is not involved. Okay,
Mm.
so there is a mixture. So we have to separate that mixture. Okay.
Okay.
For that pass through a column with immobilized oligodeoxythymine. Okay.
Okay. Okay.
When mRNA with poly A tail are retained. We
Okay.
know what is poly A tailing mechanism?
Post-transcriptional tailing.
Yes.
Yes.
Tailing in which end? 3 prime or 5
Three.
prime?
3'.
5 prime what happened?
Capping.
7-methyl guanosine capping.
Capping.
Capping is
Yes, ma'am.
there. So
Yes.
3 prime and AAAA. So at that time we discussed this poly A tail act as a signal.
Yes, ma'am.
Okay, this we can use this poly A tailing as a signal molecule. Okay, so we pass this rRNA and mRNA through a column. That column contains immobilized oligo DT.
Yes, ma'am.
Okay, what happens is that this particular mRNA contains 3 prime poly A tailing.
Yes, ma'am.
Adenine always binds with what?
Thymine.
Thymine. So we can easily remove which one? We can identify mRNA from rRNA.
rRNA. Yes,
You
ma'am.
got the point? Yes.
Yes.
mRNA is eluted. These are different sizes coding for making different protein. So we can fractionate this mRNA species. Specific insulin mRNA is identified and separated by passing through the column. Okay.
Yes.
Okay. Then DNA synthesis by reverse transcriptase plus DNTP, RNA DNA hybrid is formed. Then finally, extend 3' end of DNA by terminal deoxynucleotidyl transferase. Then finally, RNA hydrolyzed by RNAs and making single stranded DNA. Double stranded DNA is formed. Okay.
Yes.
Then DNA double strand is completed by the action of DNA polymerase. Now we got cDNA. Okay. Then sticky ends of cDNA are generated by terminal transferase. You just understand how we take this RNA combination, rRNA, mRNA.
Yes, ma'am. Yes.
Then how we separate mRNA. Okay. With the help of that immobilized medium containing oligo-DT and also poly A tailing. Okay.
Yes, ma'am.
Then we fractionate mRNA, that is, these are of different size. Okay.
Yes,
different size of mRNA coding for making different proteins. Okay. So we can fractionate these mRNA pieces and we isolate specific insulin mRNA. Okay.
Yes, ma'am.
Fractional separation like that. Okay. Then next we get an mRNA. So it's an RNA. Then we start our reverse transcription. You know the steps of reverse transcription. Okay.
Yes,
so RNA is a single stranded molecule. Yes. First of all, we have to synthesize one double strand. It is known as RNA DNA hybrid.
Yes, ma'am.
You remember this mechanism? First we synthesis RNA DNA hybrid. Okay.
Yes, ma'am.
Then next we extend 3' end of DNA by terminal deoxynucleotide sequence and RNA hydrolyzed by RNAs and making a single stranded DNA. Okay. Single stranded DNA. Then we form a double stranded DNA. Okay, double stranded DNA is completed by the action of DNA polymerase. You just write down this cDNA is formed by reverse transcription mechanism. That's all. You can avoid all the complications.
Yes, ma'am.
Otherwise, it will be very difficult. DTTT and all other things. We can't complete our question also. Okay.
Yes.
So normally the questions will be like this recombinant DNA technology. So when we write all these points, there is no need. We are not specializing molecular biology and all.
Yes, ma'am.
So no need. How can we fractionate this mRNA? How can we synthesize this cDNA? That's all.
Yes.
Okay. So we complete that particular topic, which is the topic preparation of. It's a procedure. Actually, we are going through the procedure of DNA recombination and we complete our preparation of specific human gene. Okay.
Yes.
Now it is the time for your question. How can we prepare specific human gene or
Uh,
cDNA preparation?
yes, ma'am. There's first we start with
Mm,
mRNA.
mm,
There is for example insulin.
mm,
mRNA will that insulin mRNA insulin protein will be
mm,
abundant in beta cells of pancreas.
mm,
So that mRNA they'll have only coding sequences
mm,
because during transcription,
mm,
the post transcriptional changes, the
mm,
introns are removed and only
mm,
exons are present.
mm,
So from the specific mRNA, cDNA is
mm.
produced by reverse transcriptase.
Okay, then how can we prepare the cDNA, preparation of cDNA molecule? Hmm?
Uh,
From
First,
mRNA.
yes, ma'am. First, there will be both containing mRNA and rRNA.
Mm,
To differentiate between them, they're immobilized with oligo DT
mm,
and with respect to poly A tail in
mm,
mRNA.
mm,
So, mRNA is eluted. That
mm,
is, there are different sizes coding making
mm,
different proteins. Then
mm,
insulin is identified.
mm.
Uh, then cDNA is resynthesized through reverse transcriptase mechanism.
Reverse
For
transcription. Okay.
yes,
Then can you explain that reverse transcription?
uh,
Hmm?
first there is a RNA DNA hybrid
Mm,
is produced.
mm,
Then there is extent in the 3' end of DNA.
mm,
Uh, then a hydrolysis of RNA will take place by RNAs.
mm,
Then uh, there is a primed by oligo DG. There's many G guanine containing at the 5' there is going to be priming.
mm.
Then double-stranded DNA is formed by
Okay.
DNA polymerase.
You can just write down virus DNA, single standard. You can just write down the points. I will give you in a short form. Okay. Hmm?
Yes.
You can just write down. Okay. I will give you. You just add the points of reverse transcription along with the cDNA. Okay.
Okay.
Actually, I'm not adding. It's a very complicated mechanism. Otherwise, you can't remember. You will be confused after 1 month or 2 months
Uh,
when you
uh,
take this note. What happened? I can't understand. Okay. That's why.
yes.
Okay. That's why I will give you small points. Okay. You can write it down.
Yes, ma'am.
Okay. So, single stranded mRNA
Mm.
hmm, plus
Yes.
plus
Plus
primer RNA.
primer RNA.
Okay, primer RNA, the requirement of primer RNA for the synthesis of DNA
Synthesis of
strand.
yes, ma'am.
Okay, then you can just give an arrow to
Down.
downward. Okay. Hmm. Then the name of enzyme polymerization by reverse transcriptase.
Name of enzyme polymerization.
Polymerization by reverse transcriptase.
Yes, ma'am.
Okay, then within bracket you can write what is this reverse transcriptase? RNA dependent DNA polymerase.
Mm. Yes, ma'am.
Okay, when we I enter into another point. When we write down one answer, okay, it should be clear. All points should be clear. You must teach that teacher, okay,
Okay.
with all your points. Hmm?
Yeah. Yes,
Then
ma'am.
only we can feel, teachers can feel, okay, she knows
They
something.
know.
Okay.
Okay.
Otherwise,
Yes.
some people, some students will write all diagrams, more confusing. Okay.
Yes, ma'am.
When we study one topic, it should be clear.
Yes, ma'am.
Okay. That's why I give you this point because I feel that much confusion in that diagram. Okay. Okay.
Yeah. Yes, ma'am.
Then, reverse transcriptase is otherwise known as hmm?
The RNA dependent DNA polymerase.
Okay, very good. So the product is one RNA and the next strand is DNA.
Product
Okay,
is
is it clear?
product
You
is
can
one.
write down. You can write two strands. One strand of RNA and the second strand is DNA.
Yes, ma'am.
Then you can write the name of that strand RNA DNA hybrid.
Yes, ma'am.
Okay. Then you can draw one arrow to downward direction. Okay. Then the enzyme name is RNase H.
RNase H. Okay. Yes, ma'am.
Then right, that hydrolyzes RNA.
Mm.
Okay. Now we have only one strand.
Mm. That
Yes.
is DNA. Yes, ma'am.
Yes. Very good. DNA copy of RNA. Actually, it is the
Ah.
DNA copy of RNA.
Yes, ma'am.
Yes.
Mm.
Is it clear?
Yeah. Yeah.
Yes.
Yes, ma'am.
Then next you can write one more arrow to downward direction and then write the name DNA polymerase 3.
Yes, ma'am.
Okay. Now we can write two strands double stranded DNA.